known by people who are not working in this field, like what is the genome, what is the structural genomics and the functional genomics. So consider that in every cell, living cell, so every living organism has an instruction manual that is into the cell, that is the DNA, in which is written exactly how that organism will be made and what it will do and how. We can say that all the information is written in a genome that is like a pile of books, like what we call in Italiano enciclopedia, and that corresponds to the genome. So the genome is the entire set of DNA that is part of a cell. And then every book is a chromosome. So we can consider that inside our cell there are several chromosomes, so there are several books carrying the information. And the information is a single instruction that we call gene. A gene may be like the color of your eyes, the color of the ears, and so on. So everything we are is defined there. Okay? We define as a structural genomics the study of the genome. So when we do not know an organism, we first try to understand the genome. So we try to copy all the genetic code that is written in the DNA. Only later on, when we have the entire set of information written in the genome, we try to understand the gene. And this is the most difficult thing, okay? Because the book of life, this instruction manual, is written in a language that is very much difficult for us to understand. It's like reading something that is written in a language, in an ancient language. So it takes a lot of time to understand what is written there, more now than obtaining the information that is written in the genome. So consider that in 25 years of molecular biology, we have been able to understand the function of a very small number of genes. And it will take a very, very long time to understand everything. So we need some tools that allow us to focus on the genes that we believe are more important for the phenomenon that we want to study, in order to dedicate all the energy that we have to understand what those genes that are candidates for us are. And we have a tool that allows us to understand which are the most important genes that we have in a certain organism. And I'm giving you here an example with plants, but I'd like to give you another example by telling you, if you look at my cell, or in any of my cells, there is the same genetic information. So the instruction manual is the same in every of my cells. But if you look at the skin, and you compare the cell of the skin with the cell of the liver, or of any other organ, you realize that they are very much different one from the other. So they have the same genetic information, but they are damn different, because every cell uses a certain amount of instructions that are into the catalog. And today we have technologies that allow us to see which information the cells are using in order to behave like it is. So here there is an example of mast cell cells and pancreatic cells. And here the name of some genes, probably the people who are at the end of the room cannot read it. But I think it's not difficult for you to understand that insulin is not produced by the mast cell, it's produced by the pancreas. So for sure you will find the genes that are involved in the synthesis of insulin in the pancreatic cells, and the information that is required to a mast cell to function will be activated in the mast cell and not in the pancreatic cell. So we can look.
Básicamente, entonces, por las dudas que la parte esa no se hubiera entendido, él dice todas las células de su cuerpo son iguales y tienen el mismo ADN, todas, nada más que expresan diferentes genes en diferentes partes de, por ejemplo, en el hígado se expresan diferentes genes que en la piel. De ahí que hay diferencias, pero el genoma es el mismo, es el único y es diferente que el de cada uno de las personas, pero dentro del mismo organismo hay diferentes expresiones génicas, ¿no? Que ahí es donde entra lo que se expresa o se transcribe, o lo que se llama la transcriptómica, ¿no? Genómica, transcriptómica. Solo para los que no entienden tanto el inglés, quizás redondear un poco el concepto.
Gracias, Francisco. Just to make you understand how complicated is this kind of study, we first have to do genomics, and so we first have to identify the DNA and to read the DNA of a single organism, and then we have to understand what there is written there. And just to read the genome, consider the effort for sequencing the human genome. To sequence the human genome, it took 13 years to a consortium that was made by groups all over the world. Italy did not participate because we didn't have any money. And we paid that gap for several years, because by not participating to that project, we did not build a new generation of genomicists in Italy. Then we have been lucky, because with the grapevine, we have been able to get into the genomic business and to start also teaching to our new people this technology. And also look not only the time required for this effort, but also the cost, $3 billion to sequence the first genome. The information that we took out of it was very much important. We found that so the Neanderthal is part of our DNA. We have between 1 and 4% of the information coming from Neanderthal. So for sure there has been some cross between the two species. And the number of genes, so the number of instructions that we have, that was estimated to be about 700,000, with the sequence of the human genome.
700.000 genes, ¿no? Era calculado, estimado. En realidad,
well, now, after the genome sequence, we realized that in the human genome there are 22,000 genes.
22.000 genes en el genoma humano hoy, ¿no? Reales.
So you understand the importance of having the sequence. Without that, we could not get information about the genes that are in our body. And if you don't understand how the machine works, it's very difficult to find something to repair the machine, right? When we are sick. Or in the case of other organs, how to improve. So the time required for sequencing and the cost so high for sequencing the human genome was due to the technology that was used at that time, the sequencing technology that was invented in the early 70s, and one of these instruments cost around $200,000 and is able to produce a very small amount of information. So in order to decode one human genome, this instrument requires 15,000 days. So you can reduce the time by buying more instruments, but you have to spend a huge amount of money, okay? And so because of that, you realize that very few centers in the world had the technology to sequence a human genome until a few years ago. And because of that, you can see in this slide the gap between the first human genome sequencing and the second. The second human genome has been sequenced 7 years later in 2008. So it has been like going to the moon, you know. First time you do your best in order to go there, but before to go there the second time, you try to improve the technology and to reduce the cost very much. And in these 7 years, the technology improved very much and is still improving dramatically. So with the new technology, it was possible to sequence the first real single individual, because the human genome is a mix of nice guy, bad guy, happy person, bad person, brown, blue eye, and so on. It's a mix of information in order to have a representation of the human genome. Because I thought that my voice was strong enough. Okay, so you can see already 7 years later, 2 months and $2 million compared to the 13 years and $3 billion of 7 years before. And now, this is a slide of last year. In 2010, this is the second case of a patient with a disease that has been sequenced in order to identify the reason for the disease. This baby boy had 100 surgeries in just 1 year, no hope. At a certain point, They decided to sequence it, they found the mutation, and this guy is now going at school. So they solved the problem in 2 weeks with about $30,000. So you see an astonishing improvement of the technology.
O sea, en 10 años pasó de un costo inviable a un genoma humano hoy cuesta menos de $30.000.
Today it costs less than $10,000.
O sea, uno podría secuenciar más fácilmente a la medida que la tecnología sigue evolucionando.
Verona, that was the introduction part. I now move into the grapevine part. I'm coming from Verona, this is Italy and this is Verona. It's very close to Venice, 1 hour by train, so we have the Romeo and Juliet balcony, it's a fantastic city, and we also have this fantastic wine. A bottle of this wine, Amarone, can cost $500 or 500 euros, so more dollars. So it's very much expensive, less known probably than the Barolo, but I can tell you that when Berlusconi was not doing something strange, that was doing his job and visiting presidents of other countries, he was bringing bottles of Amarone as a gift, not as a Barolo, because this is our most important, I would say, and good wine. It's made through a very particular process that we call appassimento. So we leave the berries for 3 months on a shelf in order to rest, and during this period, this terrible grapevine produce a fantastic product. So if you make the wine without this period of withering, the quality of the wine is very bad, or actually it's not good. After 3 months, it's spectacular. The problem is that winemakers, they don't know why the wine is coming so well.
O sea, él lo que quiso decir ahí que la producción del Amarone es muy trabajosa porque hay que secar las uvas al aire, bajo techo, pero al aire, y eso hace pasitas que permiten después hacer un vino más concentrado, que es un producto muy caro. Él dice que solo lo supera los Barolo, Barbaresco, que es la variedad Nebbiolo en el Piamonte, pero que por ejemplo Berlusconi prefiere regalar a Amarone cuando va al exterior. No sé qué le habrá encontrado a Amarone. Entonces es un ejemplo que él da de un vino que puede costar 500 euros la botella, por ejemplo, un Amarone bueno, muy famoso.
Anyway, Verona, that was around 2005, okay, a few years ago. The winemakers were already afraid that they had to compete on the international market. And the competition, they expected to become stronger and stronger every year. The problem of the competition is that now several countries are using the best technology. So it's very difficult to improve a product just based on the technology, because technology is already very high. The only way to improve the quality of the product at this point is to improve the initial product, so the grape wine. The problem, and this is Giorgio Pasqua, a winemaker from Verona, one of the biggest producers of that area, kept telling us, you know, Massimo, the grape varieties are more than 200 years old. There is no breeding on that in this field. So we keep using the same varieties, but the problem is more the fact that, you know, for us, producing good wine is more an art than science. So producing a good grape is not based on science. So we should get more information about how to control the engine, so how to control the grape during berry formation. And because of that, four winemakers, Bolla, Masi, Pasqua, and Sartori, one of the four more important winemakers of the Veneto area, that is a very important area for wine production in Italy, decided to give us money and to start a pilot project. They didn't give the money alone, because they were giving us money also. The Veneto region gave us money. It's always like that. If no one starts, you don't get anything. But if there is somebody who sees something, at the end the other, usually the government or the public institution follow. So with that €1 million, we have been able to go to the Ministry of Agriculture and convince the Ministry of Agriculture that it was the time to start a genomic program in order to understand grapevine. And we started also discussing and talking to the French partners, and at the end we have been able in 2006 to sign an agreement between Italy and France in order to sequence the Vitis vinifera genome. We did it in 2007. I just one of a number of groups who participated in that initiative. I have to say that in Italy at that time we were very weak as a genomicist. So the majority of the work has been done by Genoscope, so by the French partner. Okay? But,
básicamente entonces él lo que explicó es que la industria de Verona fue un poco la semilla para que se haga la secuencia de Vitis vinifera en el mundo, porque no existía secuenciado a la Vitis vinifera. Y eso nace con la iniciativa de ese grupo de 4 bodegas que invierten un porcentaje en la Universidad de Verona para comprar e instalar un equipamiento de genómica en la Universidad de Verona. Y entonces Verona se asocia con Francia, con algunos laboratorios que ya estaban en Vitis vinifera pero que nadie había secuenciado el genoma. Y esta es un poco la publicación inicial, el primer genoma de Vitis vinifera que eligen el Pinot Noir porque era... Why you select the Pinot Noir?
Because we wanted to sequence the Italian variety. The French people wanted to sequence a French variety, so we decided to sequence an international variety.
Así que decidieron por el Pinot Noir, o en Italia le llaman el Pinot Negro, como una representativa más internacional que, por ejemplo, si fuera la región del Amarone, ¿no? Entonces iniciaron el proyecto con eso. Él va a explicar ahora que después pasan a una segunda etapa donde secuencian la corvina del Amarone, ¿no?
And anyway, you can see the time that this project required and the cost, 13 million euros. So it's very much. But the information that we got out of this sequencing project, they were very much important. For instance, we found that in grapevine there are more genes than in human. So it doesn't look like, but grapevine is more complicated than what we are. And to understanding how grapevine is functioning is probably complicated as much as it is complicated to understand how we, as a human being, we function. Okay?
Él explica entonces que el genoma de la Vitis vinifera tiene 8.000 genes más, vieron, de 22.000 genes que tiene el humano, tiene 30.000, lo que lo hace más complejo, el genoma de Vitis vinifera. Lo que también lo hace interesante porque eso significa que puede haber más diversidad o más variaciones. Y en definitiva que en ese cuadro que él mostraba estábamos entre el humano está entre el pollo y la Vitis vinifera. Vieron el chicken y la uva.
And I have to say that with this sequencing, because we released the genome, of course, but the expertise that we have been able to accumulate during those 3 years, at the end were very good for us because we learned a lot about grapevine and we became the reference centers for grapevine all over the world. So we have now people from the US coming to Italy to learn how to analyze this genome. People from Chile, people from all over Europe, they now come in a place that until 5 years ago didn't know anything about genomics and knew very little about grapevine. We were very good winemakers, but we were producing that because of the art and not because of the science. So during the last years we improved our capacity for analyzing genomes. I'm also sequencing human genomes. So that is it's quite strange because the expertise that we accumulated on a grapevine at the end is so good that we can also analyze human genomes with this instrument that was used also to sequence the corvina genome because the winemakers once again came to us and told us, okay, you guys have been able to sequence an international variety, but we do believe that every variety is producing a different wine. So the soil is very much important, the environment is very much important, but also the variety is very much important. In Italy we have 300 varieties, and we must be able to highlight the quality of each of those varieties. The first one we really want to characterize very well, because we need it, is the Amarone. So because of that they injected another 300,000 euro that together with the Veneto region co-founding raised the grant to 500,000 euro to sequence the Corvina genome. That was 3 years ago. Because of that, we sequenced the Corvina genome, understood the differences, structural difference compared to the Pinot Noir. There were some duplications, so some genes were duplicated, and we found several variations. These are the DNA polymorphism. There are around 400,000 differences between the Pinot Noir and the Corvina. And also the Corvina is very much heterozygous, something that is known in grapevine.
Esto es el ejemplo de la Corvina, entonces la variedad típica del Amarone, y recuerden este número que está a la izquierda, que son casi 400.000 variaciones en el genoma de Corvina con respecto al Pinot Noir, porque él va a mostrar ahora los datos del TANNAT, ¿no? Entonces está bueno que se recuerden de esta diapositiva.
Since the plant genome is not a mix of different genomes, I told you the human genome is a mix of several persons who donate their DNA. So everything is represented there. But the grapevine genome is the Pinot Noir genome. So we were not sure that all the information or all the biodiversity of grapevine was represented in the Pinot Noir genome. So we decided also to follow the approach that has been used in 2010 to sequence the panda genome, and so to do de novo assembly of pieces of DNA that were not matching with the Pinot Noir genome. And by doing that, we found that Corvina has a set of private genes, genes that are present in this variety and that are missing in the Pinot Noir. So that's opened a new scenario, because probably every variety has a particular set of genes that is making the difference, and between us the number of genes is more or less the same. So the differences between us are based on those small differences that are reported there. So here we have a higher genetic diversity that is probably really very much contributing to the characteristic of the wine that is produced, for instance, by this plant. And there are also 3,000 genes that are used by the plant only during this particular phenomenon, and that are very much helping the winemakers of Verona to understand this process. So 2 years ago I was here invited by Fernando Alvarez for a course of bioinformatics in Montevideo, and had a few hours to go around. I asked Fernando to organize a meeting with my best friend and one of the most important person in the world working on peroxynitrite, Rafael Radi. I've been able to visit at his place here, and also to meet Francisco. Somehow we met, and somehow we started talking about the TANNAT. I was not aware of what TANNAT was. I never heard about TANNAT, I admit, probably because in Italy we have 300 varieties. I was not aware of this one. He described me this one. Actually, we spent a couple of nights also drinking very good TANNAT. So I went back to Italy convinced that TANNAT was a very good wine, and that understanding the
We need a lot of bottles. Nos gastamos unas cuantas botellas para convencer.
So anyway, we have been in touch, and last year we started the project that led to the sequencing of the TANNAT genome. We got a very nice coverage of the genome. We produced very high quality data, and with this data we have been able to find some very important duplication. I will talk later about the functions of this duplication and the genes that are involved in this area. I just like you to try to, and you Francisco, if possible, to explain in Spanish this point that for me is very much important. So I was comparing before Corvina with Pinot Noir, and I told you that there are 400,000 differences between the two plants. By comparing Tannat with Pinot Noir, the differences are almost 3 million. So I don't know if that is because Tannat came and because the variety that was sequenced is a very old variety, one of the first clones that came here, right, Francisco?
Yes.
But probably because of that, I mean, humans can cross the ocean and mix the DNA, but plants cannot do that. So those plants have been on the Uruguayan soil, separated from Europe for such a long time, that this may be an explanation of this dramatic difference. So heterozygosity is still high, but the differences compared to the Pinot Noir are really astonishing.
O sea que comparativamente con lo que él dice, que tiene casi 3 millones de bases diferentes en los genes, el Tannat con respecto al Pinot Noir, y ustedes se acuerdan que en la Corvina del Amarone había unos 390.000, o sea que esto tiene mucho más diversidad genómica que la Corvina y que el Pinot Noir. Y él piensa que esto puede deberse a que es una variedad antigua, que puede verse también, nosotros sabemos que es considerada por algunos genetistas el Tannat como una variedad muy cerrada, o sea que no tuvo cruzamientos con muchas variedades. Por eso él da el ejemplo, los humanos podemos cruzar el océano y nos cruzamos con un africano, europeo, lo que sea. Las plantas no pueden cruzarse, entonces el genoma de las plantas es más diverso, digamos, que el que está cruzándose continuamente dentro de la misma especie. Y entonces esto muestra un primer panorama del polimorfismo del ADN del Tannat, que es muy rico, lo cual eso es una buena noticia, ¿no?
Yes, because in Italy they say that a wine to be recognized must be recognizable. So it must be different somehow. And here I have to say the variation, not always to be different means to be better. Okay? So, but for sure in this case the differences genetically encoded differences between Pinot Noir and Tannat are very much high. Anyway, so we then continue the work because sequencing is giving you